your inner membrane, this is your phospholipid bilayer, your inner membrane. You would have your peptidoglycan layer, this thin layer here, and then you would have another outer membrane. Now the thing about this outer membrane is that one of the leaflets is going to be a phospholipid layer, just like the other membrane. But the outer leaflet is actually going to be composed of a completely different molecule. Stay with green, I guess. I need another color. This outer leaflet is a lipopolysaccharide. It too is a uniquely bacterial characteristic. So now we've identified two molecules, peptidoglycan and now lipopolysaccharide or LPS, that are uniquely bacterial. The reason I bring that up is if you can develop drugs that target just those molecules, then we don't have to worry about our cells being damaged by those drugs. Anyway, so we have this outer membrane here with one leaflet composed of a lipopolysaccharide. When we look at the parts of this lipopolysaccharide, what we see is that this outer structure sticking up here, this is called an O-specific molecule. An O-specific polysaccharide, and it is an identifier used for the identification of the cell. We can identify cells based on the type of O-specific molecule they might contain. In addition to that, this molecule here is known as a lipid A molecule, and it is an endotoxin. The presence of that molecule on gram-negative bacteria elicits an immune response from your immune cells. A small amount of lipid A would elicit the appropriate immune response, but if you had these bacteria actually multiplying in your bloodstream so that you had a much higher concentration of lipid A, the immune response might be so great that it actually starts damaging your own cells.
So the lipid A is just the little tails, the double tails off the
It's actually this whole structure right here. Yeah, it's this whole structure right here. And they can vary. You'll see in your book that they vary them with various structures coming off of the little square. But just know that this here structure is the lipid A, and it serves as an endotoxin. So, and here my point is with regards to the term endotoxin, means that the microbe itself, or parts of the microbe, are the toxin, as opposed to one that's being released. We talked about Staph aureus releasing a toxin, or Clostridium releasing a toxin. Okay, those are exotoxins that the cell is secreting. This is an endotoxin because it is actually part of the cell and elicits an immune response when exposed. So, gram-negative cells, they're actually, gram-positive cells, actually very simple when you compare them to gram-negative cells. They're just an inner membrane and then many layers of peptidoglycan But then when you get to the gram-negative cell, you have your inner membrane, your phospholipid bilayer, one or maybe two layers of peptoglycan, and then you have this outer membrane. Any questions?
Like osmosis is for water movement, what's the other one?
Diffusion? Diffusion is the movement of things that's not water against a gradient or with a gradient.
Does the outer membrane also have proteins in it?
Yes, good question. It does have proteins in it. And those proteins can serve in the movement of molecules across that membrane.
So molecules would move through the outer membrane, through the peptide membrane.
Yeah, this really is not going to be much of a barrier, really. It's more of a rigid structure like a lattice work. It's not really going to stop a lot of stuff from moving through it.
So the peptide layers are all like this? Or no, they're not.
They're like this.
Oh yeah, that's right.
Okay, so one layer, so this is one layer here.
Yeah. Yeah.
So you're saying cell wall also can have channels, protein channels?
Yes, can have proteins embedded into the membrane. Not the peptoglycan layer, but the outer membrane can have proteins embedded in it as well.
So it has a channel in that lipopolysaccharide layer, but it won't go through the second membrane.
No, it probably will. There probably is another channel.
It passes that matrix of the peptidoglycan.
I'm sorry, what was that again?
I think I misunderstood you. You said that there won't be a channel in the peptidoglycan.
Because this is like a lattice.
But that protein channel goes straight through that lattice.
No, it won't. The protein channel will just allow the molecules to pass through this layer of membrane.
Okay.
Then they'll pass through the peptoglycan layer, and then they'll probably have to pass through another protein channel or something else to get into the cell.
So they diffuse across the peptidoglycan layer.
Yes, you could say that they diffuse across that if you want. Yeah. Okay, so some compounds interfere with the formation of peptoglycan. Penicillin is one of them. That's why penicillin is very effective in treating certain infections. However, it's going to be effective in treating gram-positive infections. Penicillin not as effective in treating gram-negative microbes. We have lysozyme, which is a molecule you'll find in our tears and sweat and things like that. Also effective against gram-positive cells, not so effective against gram-negative cells. This outer membrane can provide some protection for the cell against some antibiotics. It's less sensitive, if you will, to some antibiotics. One more thing that we can say about peptoglycan, archaeobacteria don't have peptoglycan. They can have pseudopeptoglycan, some molecules that are similar, but only bacteria. And this is what defines the group really, have peptoglycan. Remember cyanobacteria, I talked to you about cyanobacteria once being thought of as blue-green algae being part of the algal group. Well, when we determined that they had peptoglycan in their cell wall, we couldn't leave them in that group anymore. We had to move them into the bacterial grouping, classification. Okay, so back to our cell that we're building here. So we've got a cell membrane, we've got our cell wall. You'll also find that there are surface layers external to the cell wall called the glycocalyx. And the glycocalyx is outside the cell wall, secreted by the microbe itself. And we can call it a couple of things. If it's entire and complete, we refer to it as a capsule. If it's diffuse and irregular, We call it a slime layer. Really, what we're talking about here is a gel-like layer that kind of helps the microbe stick to a surface or help it stick to another bacterium. Your biofilms can develop because of the slime layers that the bacteria have, allowing for many, many bacteria to stick together. In addition to that, the glycocalyx can also protect the microbe against cells that might want to destroy it. For instance, some of our immune cells. The glycocalyx can serve to mask the bacterium. Bacteria have a flagellum. This flagellum generally has 3 parts, but I won't worry about the 3 parts for the most part. Too many G's in there. Now, some microbes, some bacteria will only have the 1 flagellum, and others may have many flagella all over the body. If they have many, we refer to them as being peritrichous. It is the flagella that allow the bacteria to move. And bacteria move by sensing their environment. Actually, I'm going to stay on this board here and just erase this stuff there.
Professor Hayes, that word you just said with many flagella, what, can you spell that?
Peritrichous? Yeah, I can spell that. Well, maybe I can't, but I'll try my best here. Peritrichous, just meaning lots of hairs coming off of it. Lots of flagella.